Good evening. So I'm Donnie Huang. It's my great honor to introduce our speaker tonight, Professor Sally Mcfadden. For those who do not know her, Sally has been in the myopia field as a luminary for over 35 years. She started off in Canberra at the Australian National University, in which she got a Bachelor of Science in Pure Mathematics, as well as in Neuropsychology. She then continued on at ANU to get a PhD in neurobiology. She then moved on to University of Newcastle, where she was for over 3 decades, with small stints at the University of Paris, Cornell University in New York, as well as the City College in the city of New York, City University of New York, as well as in Barcelona as well. I've known Sally myself for over 10 years, and I was fully impressed the first day I stepped into her laboratory. Clearly, she was a basic scientist, but she had everything you can dream about if you're an optometrist or an ophthalmologist, but all for guinea pigs. And I really do think this is the main draw of this great talk of mechanisms underlying myopia, because even for my own daughter, I'm now thinking about different lenses that can control myopia. I have a 9-year-old and a 4-year-old, both of which are myopic already. But only with a true understanding of the mechanisms behind myopia can we truly understand why these different myopia treatment modalities exist, what science led to their discovery, as well as really, if you're talking to patients, how you choose one over the other. Only with the proper understanding can we do that. So with this, I turn it over to Sally. Oh, Sally, I think you need to unmute yourself.
Thank you for that kind introduction, Donnie. So we all know that if in ideal conditions, if the images are focused at the back of the eye, then that's wonderful. But in a myopic eye, the eye has basically elongated and grown too fast for the optics of the eye, and so that images are short focused. And of course, we can correct that with negative spectacle lenses, but that doesn't change the fact that the eye's already grown too elongated. And the problem emerges if that continues to happen, so that the eye develops high myopia, and then there can be blinding pathologies that develop, such as separation of the retina from the back of the eye, or these bubbles that form in the back of the eye, staphyloma. And so the issue really is that the prevalence rates have increased dramatically in the last series of decades, and unfortunately, we're still on track for this data, even though we've now developed a lot of technologies in order to try and slow the progression of myopia. In reality, half of the world's population is still likely to be myopic by 2050, and of those, nearly 1 billion people will have high myopia. And to put this in perspective, that's more people than caught COVID during the pandemic between 20 and 24. So this is obviously a big pressing problem. So what I want to talk to you today about is introduce you to the animal models and tell you a little bit about what we know about the myopic signals and how they're produced within the eye. And we still don't know the answers here, and there's a lot of research that's still undergoing. So if we look at the animal models, there are a lot of different species that have been used to study myopia, but they all have one thing in common, and that is that the eye grows during development, and during that period, the eye emmetropizes and loses its refractive error that it starts out with. Most species are hyperopic at birth, except for the emu or the ostrich that have huge eyes. But the refractive error dissipates over time. So that's similar across all species. And the underlying mechanisms that I'm going to talk about today are amazingly phylogenetically conserved. So I developed this guinea pig model that Donnie was just telling you a little bit about. And it has the advantage, these animal models here have the advantage that we can manipulate vision during this early growth period and try and understand what it is that causes the changes in the eye. And we can take an animal and manipulate the vision just for a week, and that would be equivalent to doing changes in a human or a monkey for over many years. So the first way we can induce myopia experimentally is using form deprivation, and this occurs naturally in children who are born with cataracts or have drooping eyelids. And in fact, the first monkey studies that were done in monkey had eyelids which were closed, and they saw that these animals developed myopia. But Josh Warman and colleagues in New York developed an easier way to do this in young chickens, where they put a diffuser in front of the eye and degraded the image, and then those animals become highly myopic. We can also change that diffuser to just a more detailed kind of filter in front of the eye, where we deliberately manipulate components of the signal to try and understand what it is that's causing the eye to go myopic. And so in our own model here, in the form deprivation model with guinea pigs, where we manipulated the images to the eyes by wearing diffusers during this sensitive period of development while the eye is emmetropizing, you can see that even after 6 days that the eyes wearing the diffuser becomes highly myopic, compared to the other eye, which was just didn't have anything over wearing, didn't have any changes, and wasn't wearing a diffuser. And this can be accelerated and increased if we continue the form deprivation. And you can see that after a couple of weeks, the animals have developed 7 or so diopters of myopia. So we can get very high myopia in these animals over a very short time period. So if we then do the other method by just manipulating the image here, here's a kangaroo where we see high detailed image, and then if we gradually remove the high spatial frequencies and degrade the contrast, you can see what's happening to the eye growth as the eye loses that detailed information. And this is the difference between the eye that is seeing the normal images, for a guinea pig that is, versus these degraded images. We're not putting them in rooms with kangaroos, by the way. And then at the same time, of course, as the eye's growing longer, then the myopia is developing. And you can see that there's a very nice relationship between how much myopia develops and how much the eye grows relative to the kind of image that we're putting on the retina. So the good news is that we can readily get recovery from form deprivation myopia if we interrupt those signals. And we only, the inhibitory signals, if we just remove the diffusers and give whatever is normal vision for even 30 minutes a day, is enough to halve the amount of myopia. So the inhibitory signals, or the signals that stop the myopia from continuing, are very potent and they're quite transient. Whereas the signals that underlie the production of myopia in the first place are quite slow and sustained, and they summate over time. And so if they're left undisturbed, they can be sitting there in your eye for days if you don't interrupt them with one of these inhibitory signals. So that's the good news, that we only need 30 minutes a day, but of course that only halves the myopia. If you really want to get rid of all of the myopia down here, then you really need to have 6 hours a day of normal viewing. And so the issue is, how can we get these signals down here to be more efficient and more effective, so we don't need to have 6 hours a day? Because the reality is that our urban lifestyles are such that we live in a myogenic environment. So one possible way is that we can add, for example, blue light onto the signal, and that might help shorten the time frame. But they're still relatively long time frames, and so that's one of the purposes, is to try and understand these inhibitory signals to help that process. So the other method of inducing myopia experimentally is to use either spectacle lenses or contact lenses and put defocus onto the eye. And obviously the more defocus you put onto the eye, the less the capacity and the resolution capacity of the eye, and the number of cycles per degree, or the spatial frequency, is reduced as we increase that defocus. And this works by putting, for example, a minus lens in front of a growing young animal eye, and you find that that will put the image behind the retina. And what the eye does is it accelerates its growth in order to compensate for that lens. So it's all fine while the lens is in place, but when you remove the lens, then the eye is too long and now myopic. Conversely, if you take an eye which is already myopic, or if you put a plus lens on a young eye, and make it give it myopic defocus like this, the eye realizes that it's got a myopic defocus signal, and it inhibits its growth now, and it does this by slowing down its growth as well as expanding the choroid, which is the vascular bed between the retina and the sclera. So these inhibitory signals are relatively rapid and potent, as I mentioned, whereas the growth signals are relatively slow and sustained. So as an example of that spectacle lens compensation in our own model here, you can see that here are the eyes that are shrinking in terms of their length, compared with ones that are raised with minus lenses, which are growing, and there's a nice relationship. So the range of spectacle lens compensation here varies from species to species. So in primates, In humans and in guinea pigs, it's around about between plus and minus 5 diopters. In chickens, it's much larger than that. So what are we doing with these two models? Basically, we're manipulating the contrast sensitivity function of the eye. And here you can probably see the stripes, the stripes in this grating here, looking something like that, depending on how close you are to the screen at the moment. But you can see that when the contrast gets very low, you can't sort of discriminate the high spatial frequencies. And so if we deliberately reduce the contrast in the image, then you're going to give the retina a much poorer signal so that it can't see as much spatial frequency or contrast information. Similarly, if you put defocus in the spectacle lens model, then you're basically also putting defocus onto that image and creating poor images for the retina, so it's very hard for the eye to know what the signals are and what it should do. Both of those signals will cause the eye to become myopic. So what do we know about the environmental myopic signals? Well, it's long been known that near work is highly correlated with myopia. And indeed, if we live in small homes with high density, that's also associated with greater levels of myopia. And during the pandemic, the prevalence rates of myopia went up around the globe. So myopic environments seem to exist in our modern world, and we know that outdoor times is protective. And so at least it delays the onset of myopia. It doesn't necessarily help as much with the progression, but it certainly delays the onset of myopia. And so these outdoor environments, it's something to do with the light outdoors, because we know, of course, that the spectrum distribution for daylight is very different to the LEDs that we now live with indoors. And they are different again to the incandescent lights that we used to have. So there's something about the light in the environments. And so, of course, experiments were immediately done in animal models to try and see what aspects of the light were important. And in these experiments here, different species were raised with different monochromatic light. Now, in this case, in monochromatic light, the eye can still respond to plus or minus spectacle defocus and grow or inhibit its growth correspondingly. So it doesn't, raising animals in monochromatic light doesn't fix anything really. But what it does tell us is that in normal eyes, the long wavelengths or the mid and the long wavelengths tend to cause the eye to grow across multiple different species, whereas the short wavelengths tend to cause the eye not to grow as much across multiple species. However, there's some, the system is never that straightforward because there are multiple pathways that process long wavelength light, and so in some circumstances monochromatic long wavelength light can also inhibit eye growth as well. So these experiments have been useful to know that maybe there's something about these blue signals and these longer wavelength signals that are important in the pathway. So if we look around Singapore here, and Will Miles, who is on the panel here today with us, he photographed a lot of different scenes around Singapore, and he asked the question, what is it about these natural environments or mixed urban or indoor environments that might be problematic to our eye growth? And so he looked at these particular, all these hundreds of images and looked to see what the contrast and spatial frequency was in those images. And he compared it to what it would be like looking through a diffuser. And so if there were sections of this image which were the same, giving the same signal onto the retina that looking at a scene through a diffuser would give, then we would call those areas of the scene on the retina as being potentially environmental form deprivation. And so here's an example within those scenes of what the environmental form deprivation patches look like. And if you plot the amount or the complexity of those of the contrast degradation in those images as a function of whether they're natural, mixed urban, or indoors, you can see that the percentage of the visual field experiencing naturally environmental form deprivation goes up as we spend more time indoors compared with outdoors. And so these natural environments are very rich in their complexity and their contrast and spatial frequency environments. And so the Singapore Green Plan from 2030, not only does this give us much healthier behaviour if we spend more time going into these green spaces, we also now know that it's also very healthy for potentially for our eye health. So these myopic signals are generated in the retina of the eye, and we know that because form deprivation still develops without the optic nerve. So in monkeys and mammals and chicks, if you cut the optic nerve, the eye still can develop form deprivation myopia. And indeed, if you even stop the signals going from the optic nerve to the brain using something like TTX, then animals still develop form deprivation myopia. And so it seems like it's within the eye itself. Short, then we know that the signals are processed by the retina, these aberrant myopic signals, and then from the retina, those signals are translated into the RPE and into the choroid. Now, the choroid is an interesting structure. In the chick, shown here, the choroid, it expands dramatically in response to positive and minus defocus. This was first again shown by the Warman group together with Christine Wilson, where they showed that the chick eye can compensate for spectacle defocus that's on the eye. And in fact, what it does is, and this is work from our own lab here, within 24 hours, you can see that you can get up to, like this is up 350 microns expansion in the choroid, and that's equivalent to around about 8 diopters that the chick eye has compensated for. And so that means that the chick eye can actually use the choroid to accommodate in reality. And it's a relatively rapid system. Now, in primates and humans and mammals, the choroid, we never would have thought to look in the choroid if we hadn't seen these experiments in chicks first. But in the choroid also does the same thing, but the changes are minuscule. They're not clinically relevant. They're like a fraction of a diopter. And so we don't have choroidal accommodation, of course. The other thing that's important about the choroid is that the changes both in eye growth, this is a pattern of eye growth over the day and the night cycle. The eye tends to grow mostly during the afternoon in the day and not at night. And the choroid changes because the choroid, remember, is expanding with the inhibitory stimulus here, mostly occurs at night. And so it's in antiphase to the eye growth signal. And these cycles get out of sync in myopic eyes. So from the choroid then, the signals are then translated into the sclera, and the sclera remodels its collagen. It becomes very thin and it becomes weak. And in high myopia, that's one of the reasons why you start to see bulging out the back of the eye and these changes, biomechanical changes that occur in the back of the eye. So I want to now look at what happens during the development of myopia and look at the central and the peripheral. So the optic nerve region is also sensitive to the sign of defocus, not just to form deprivation. And this is data showing the eye that's wearing a -4 lens versus a control eye wearing a plano lens. And this one's wearing a minus lens here, whereas this eye here is wearing a plus lens. And we're talking about the shape of the eye here. And if we look at the difference between the two eyes as plotted down here, you can see that the eye wearing the minus lens, just like the eye that was wearing a diffuser, expands again in this region out here, as well as in the across the whole eye, whereas that doesn't happen with the plus defocus. So a plus lens is an inhibitory response in terms of the changes that are occurring in the shape of the eye. We also know that myopic eyes have relative hyperopia. In other words, if you look at the amount of hyperopia in the periphery compared with center of the visual field, that the eye is relatively hyperopic. And that's because the eye expands out the back and becomes this prolate shape. And the same thing happens in the animal models. You can see that the red and the grey lines here are what's happening in the periphery in terms of the refractive error, and they are less myopic or relatively hyperopic compared to the myopia that's developing in the central region of the visual field. And you can see that there's this very steep increase or hyperopic shift that occurs at an early phase during this development process. We also know, though, that myopia occurs without the fovea. And this was a surprising finding. And Earl Smith did this beautiful work back in 2007 and 2009, where he photocoagulated the fovea, and quite a large lesion was made here. And yet these animals, even without the fovea, which is where, of course, all of our cones most of our cones are sitting, the animals still developed myopia, showing here over time, despite the fact that they had these laser lesions. So this is the normal form deprivation animals that don't have the lesions, and these are the ones that have got the lesions. And so not all of them go myopic, but still, the eye can still become myopic in response to the form deprivation. And the same thing happens with binocular -3 diopters. So minus lenses also produce myopia. So here's the normal myopia produced by a -3 lens. It's about the average is probably about 1 or so diopters here. And a similar effect for the animals that have got the laser lesion. They don't look like the normal animals as is shown up here in these two open circled patterns. So myopia doesn't appear to need the fovea to read the signals which are going to cause it to become myopic. So this led to a lot of interest in the periphery and how it might be involved in producing signals which might make the eye go myopic or not. And so Earl, for example, did experiments in those monkeys where he put holes in the diffusers and the animals still went myopic. And this is our work in the guinea pig showing that if we put the defocus from lenses only in the periphery of the eye, then we can still create changes this is the animals here who've got the normal vision in the middle, and yet we're still seeing increases here in this sensitive zone around the optic nerve head. If you put the defocus over the entire eye, then you're getting a bigger response, but you're still seeing a response here from having defocus in the surround. Similarly, if you put the inhibitory response across the whole of the eye, which is the red one here, you can see that this is refractive error. You get less refractive error, a hyperopic shift compared with the controls that have just got plano lenses. Whereas if you put minus in the surround, you get just almost as much myopia as you would as if you put it over the whole of the eye. So these experiments suggest then that the peripheral defocus can change the central refractive error. And this, of course, is one of the reasons why multifocal lens treatments were tested to see how they might be effective in inhibiting the flow of myopia. So traditionally, a single vision lens like this would correct the vision in the center where we do most of our viewing, but may not correct the periphery of the eye. And those peripheral signals could drive the eye to become myopic. And so now new lens designs tend to try and put that inhibitory plus signal somewhere in the periphery out of the line of sight so that we can try and get that signal to then inhibit the eye growth at the back of the eye. And we did this where we mixed plus and minus lenses in the Fresnel design. And you can see here, this is eyes which have got a plus defocus lens where the eye is becoming hyperopic, or a straight minus defocus lens where the eye becomes myopic, and those eyes grow and the other ones shrink. But if you then make a lens which has got both plus and minus around in the periphery, then you can get an inhibitory sort of halfway response from lacing that lens, if you like, with both powers. In other words, what the eye and what the retina does is it integrates the different amounts of defocus, and that can produce a reduction in the amount of myopia that you might have in the eye. The thing about these lenses, though, is that the more plus you put into the lens, the better off you are. So 100% plus in the lens produces the most hyperopic shift in the refractive error. And of course, we can't just put plus over the whole lens if we're a myope, because we're trying to correct the myopia. And for that, we need to put a correction of a minus in there so that we can see. So the trick with this lens design is to try and know exactly how much of this plus defocus we should put and where we should put it. And there are many different lens designs now in the anti-myopia lenses. The DIMS lens were the ones that were developed after the Fresnel work that we did was trying to put now that plus defocus into little lensed arrays on the lens itself. But they all basically follow that same principle, which is, what about if we put plus defocus into that lens and the trick is, of course, not to disturb and not to disrupt normal vision as much as possible. So I recommend that you read some of the new reviews that are coming out about these new lens designs if you'd like to know the difference between all the types, because there are now quite a few that are on the market. So I want to talk about the basis of the competing growth control signals. One of the most vexed questions in this field is, how on earth does the retina discern the sign of defocus? Because the blur circle from a plus lens or a minus lens, if it's behind or in front of the retina, is still a blur circle and looks similar. And so if we believe that these signals are indeed encoded in the retina, then it must be through some of these primary retinal signals. And the retinal cells can sense orientation, contrast and spatial frequency, motion or colour. And so something here is being able to be encoded to encode defocus. So it is thought that maybe higher order aberrations and orientation detectors within the retina might be a way to do it. However, these are relatively small and they occur and in some species that they have very little higher order aberrations because it depends on the quality of your optics. But there is a difference in the orientation for some of these higher order aberrations in terms of the blur circle on the back of the eye. Similarly, chromatic aberration was thought to be able to provide a cue. After all, the blue light might be short focus, whereas red light would be long focused. And so the degree of chromatic aberration could be discerned by the retina if it was comparing the short and the long wavelength channels. However, we did show that, that the eye can still respond to lenses, plus or minus lenses, even when it's raised in monochromatic light. And so this suggests that it's not just the only thing that might be used. In reality, biology always has parallel pathways where it will use multiple types of cues in order to try and discern the way the eye is working. So we don't really know what it is about these particular primary signals and how they might encode defocus. There are also extraretinal signals that can influence activity within the retina. Circadian rhythms is one that we talked about a moment ago, and also accommodation is an enduring topic that people come back to time and time again to try and understand how it might be impacting these primary visual signals in the retina. I'm going to talk about contrast and spatial frequency because we know that if we degrade that, we can instantly cause a very graded change in the amount of myopia and eye growth that develops. So we know that the way vision codes images is in terms of local contrast. And so square A here probably looks dark and square B probably looks lighter. But in reality, this is a complete illusion. There is no difference at all in the luminance of those two squares. It's just that our brain tries to look at the difference between the actual square and the surround in order to try and interpret it. Now, this illusion, called the simultaneous brightness contrast illusion, was discovered by a French chemist actually back in 1839, and we know that this occurs from the retina onwards. So the retina is the cause of this. And this half circle might look a different shade to this half circle, but it's because the retina is encoding that object based on the difference between it and its background. There is no difference between those two halves, and you can see that when we just put a plain background behind it. So I'm trying to demonstrate here that it's the contrast between local areas that the retina uses to try and encode images. And there are two ways that it can do this. It can either go through the rods and through the luminance channel or through the cones and through the color channel. Now remember that Earl deleted all of this area where most of our cones are sitting. There's something like 20 times as many cones sitting in the central retina compared with the peripheral retina. But there is more like 100 million rods in the retina, so there's far more rods than there are cones. So we know that if we lesion that whole eye, the eye can still go myopic. So that means that something out here in the near periphery is able to still drive the myopic signals. So how does it do that? Well, we have to look first at the fact that the receptive fields of the neurons in the retina are coded by having a center surround organization. And this was discovered by Stephen Kuffler back in 1953. As a physiologist, he showed that if you put light in the center of a receptive field of a ganglion cell, that the cell will fire dramatically to that situation. Whereas if you put the light in the surround, it turns off the basal firing rate of the retina. The retina's noisy in the dark. It's always firing away a little bit, and it turns off that basal activity. The opposite occurs in the off pathway, where if you put light in the center, you actually turn off the response, whereas when you put light in the surround, you turn it on. So these are the two basic fundamental channels within the retina, and it is the way that the retina codes information using these local contrasts. It's not as clean as a simple on and a simple off pathway. They interact in complex ways. However, Frank Schaefel did some beautiful experiments looking at what happens when you try and make images which stimulate the on pathway. So white text on a black background will stimulate the on pathway, whereas black text on a white background, the normal black and white text will stimulate the off pathway. And if you use these kind of stimuli, he's got evidence to show that you're more likely to invoke in humans that choroidal response, which is indicative that there is an inhibitory response in the retina. However, there's just as much evidence to say that it's not clean like that. It's not as if this pathway is only the inhibitory pathway and this pathway is only the stimulating pathway. It's never that clean in biology. But nonetheless, there is evidence to suggest that the on pathway might be pretty important for the inhibitory system. Now, whether or not that also occurs in the color channel, contrast channel, is yet to be properly explored. So the way the retina codes information, then it starts with these vertical pathways. We have the on pathway, which goes from the red and green cones through the bipolar cells, of which there are about 12 to 15 different varieties, and then synapsing directly onto the ganglion cells down here, which form the optic nerve. So this pathway here is an on pathway, and the synapses, and these are called on bipolar cells, they actually have their processes in this on layer in the inner plexiform layer, whereas the off pathway has its processes in the off layer and its synapses onto these off bipolar cells. So we have these two on and off pathways right up front in the eye. The rod pathway tends to do it a different way. It synapses with bipolar cells as well, but instead of synapsing directly onto the ganglion cells, it goes via an amacrine cell known as the A2 amacrine cell. And in most species, our rod pathways tend to be within the off pathway. So the blue cone is an interesting beast because the blue cone is more phylogenetically aligned to the rod opsins than it is to the red and the green opsins. And it's got a different evolutionary history, and it also tends to go through to the on pathway. And so there is, for example, the blue on yellow off retinal ganglion cells, which take input from the blue pathway, from the blue cones. So these are the vertical pathways through the retina. What's way more interesting and what actually produces that inhibitory surround in the horizontal cells is, in the retinal ganglion cells, is these horizontal cells. So the horizontal cells now, what they do is they take signals from neighboring cones and feed them into bipolar cell pathways and feed back onto cones themselves. In order to modulate the surround. So now the surround of the receptive fields of the ganglion cells that these are then subsequently connected to eventually end up with these, with these inhibitory surrounds based on the wiring of the way these horizontal cells interact. So they're an important lateral connection within the retina. And so our retina then is tiled with receptive fields that start to read that local contrast that occurs, whether it's around the foveal region through the color and the cone pathway, or more in the periphery through the rods. But basically, they're both doing the same kinds of contrast modulation. However, the main pathway where all the action happens in the retina to a large extent is in the amacrine cell layer. There's up to 30 to 60 different types of amacrine cells, depending on what species you're talking about and how they are categorized. So there are so many of these amacrine cells, and they do all of the hardcore processing of the images in order to get light, which is captured by the photoreceptors, into actual images, which is then passed up into the optic nerve through the ganglion cells. So there's two dopamine cells that I'm going to briefly talk about. One is the dopaminergic amacrine cell shown in blue, and the other is the nNOS amacrine cell shown in red. So like the horizontal cells, these are laterally connecting cells which have their dendritic fields spread a long way across the retina, and so they can read information from quite wide varieties, from a wide distance across the retina. And the reason we're interested in dopamine and nitric oxide is because these three substances, including retinoic acid, are very important in the retina in terms of sensing, contrast, and defocus. So we know, for example, that dopamine is downregulated in form-deprived eyes, whereas it becomes upregulated, it increases in eyes that are recovering from myopia. And the same thing happens in nitric oxide, except nitric oxide is sensitive to lens wear, both plus and minus lens wear, whereas dopamine isn't so sensitive to a system whereby the eye can see through the lenses and have some kind of feedback happening, which is what's happening with the lens wear. But nonetheless, both of these substances are sensitive to their agonists will inhibit form deprivation, and their antagonists will block the light inhibition effects of form deprivation or block inhibition of the form deprivation. They also are involved in light modulation in the retina, and so involved in light adaptation. Retinoic acid is also important. It's also a bidirectional signal like these two. Retinoic acid, if you feed it to animals, it'll make the eye grow like crazy while you're feeding it to it. If you stop feeding it, it'll stop growing, and then if you feed it again, it'll grow, etc. So it's a really important factor, and it's also bidirectional for both form deprivation and lens wear. The interesting thing about these three substances is that all three of them modulate cell-to-cell gap junction coupling. So what do we mean by cell coupling? These are electrical contacts between individual neurons. They are very fast and an efficient way in order to sort of, for neurons to talk to each other. So this is an example of a network. Here's an individual cell, I don't know if you can see it there, a little tiny red cell, horizontal cell that is coupled to all of these neighboring cells here. Whereas in this situation, this horizontal cell here is coupled to many more cells. That means that that cell can see information from a much broader surround than this cell can. And it's through these processes then that the center surround receptive fields are produced. And this was shown by Stuart Bloomfield and David Vaney in separate laboratories, where if you put nitric oxide onto a horizontal cell network that's highly coupled, it will quickly shut that coupling down and turn it off. Similarly, if you put dopamine on those coupled networks, it will shut down that excessive coupling. So that's what these two neurons then, the dopaminergic and the nNOS neurons, are these horizontal pathways which are heavily coupled to one another by these gap junctions. And so that means they can process information very rapidly from the periphery or from neighbouring areas of the retina. There are, of course, around about 18 to 30 different ganglion cell types. There's many different kinds. And one of the classes that I haven't even talked about at the moment is the intrinsic photosensitive retinal ganglion cells. Now, these cells aren't necessarily involved in image detail, and they're actually located in the ganglion cell layer, but they have their own opsin to directly sense photons of light. So in addition to the rods and cones, we have even ganglion cells now that actually have opsins that sense light directly as the light comes into the retina, and they do it with a blue pigment called melanopsin. So these cells are able to control circadian rhythms, which we talked about earlier as being important in eye growth, eye movements, as well as the pupil reflex. And in the macaque retina, it's recently been shown here by Milner et al. And Paul Gamlin that they can be switched off by red light, which is very intriguing because it may relate to the red light therapy that is being used at the moment and seems to be effective. What's important about these IP-RGC cells is that all of them are coupled to amacrine cells, so they talk to the amacrine cell layer, and in particular, the M1 and the M2 talk to the dopaminergic cells and directly contact the dopaminergic cells. Remember, these are involved in circadian rhythms and cause the dopamine cells to release dopamine. Likewise, the M3 cells, which can synapse in both the on and the off layers, can talk to the nNOS cells, and they cause nitric oxide release. And so these photosensitive cells can modulate the responses in the inner retina here and modulate the production of nitric oxide and dopamine. So in summary then, it seems like we can have two states within the retina, whereby in the inhibitory state where the cell network is relatively uncoupled, this can be created by putting either darkness or bright light onto the retina that can shut down the coupling. If we have dim light, on the other hand, we end up with very wide coupling. So this sort of stimulus here produces a very coupled retina, and the coupled retinas tend to be indicative of an eye that is increasing in its growth and is going to become myopic with low levels of dopamine and low levels of nitric oxide, whereas uncoupled retina tends to be in a state where the signals are inhibiting in growth. So why do we care about all of this? And you've listened to this talk, those of you who are still with us, saying, well, how does this relate to me and the glasses that I'm trying to get for my children at the moment, or the ones that I've got for myself? Well, if we can understand these retinal pathways that promote the surround inhibition and enhance contrast, it could be a way that we can target and design new retinal signals that would specifically stop myopia from developing right at its source. And as for cell coupling, it allows these non-imaging IPRGCs to modulate dopamine and noradrenaline and nitric oxide release from amacrine cells, both factors which are very important for the pathways for myopia control. And it shapes the inhibitory surrounds of the receptive fields and can enhance contrast. And it does allow the peripheral retina to rapidly influence and control more central retinal signals. So in conclusion, there are multiple parallel pathways that underlie myopia. And animal models are very useful to untangle the complex underlying biology. We know that hyperopic defocus, which can cause myopia, may be coded by the sustained signals that relate to poor contrast, poor spatial frequency, dim light, low levels of dopamine, and strongly coupled retinal cell networks, whereas the inhibitory pathway in the retina is coded by more transient but very potent signals that might be related to, or are related to, cellular uncoupling and nitric oxide release. So I'd like to acknowledge finally the people that have been through my lab, students, postdocs that have been involved in this work, and also colleagues from around the world who have been involved in the studies that are some of the studies that I've reported today, including Josh Warman from City College, Christine Wilsett, who is at UC Berkeley, and Eugene, Grace Lee, and Jose were involved in the nitric oxide studies in our laboratory. And Dennis from Hong Kong, who's at Hong Kong PolyU, was involved in, together with Christine and us, with those Fresnel lenses, and has now gone on to develop the DIMS lens, which you probably do know a little bit about. And finally, I'd like to acknowledge my institutional support, particularly the Department of Ophthalmology in the School of Medicine at NUS, and also the University of Newcastle, where many of these studies were undertaken, and also the Singapore Eye Institute, and my grant support. And thank you very much for listening.
Great, thank you so much, Sally. So now we're at the Q&A session. So please, if there's any questions, feel free to type it into the Q&A session here. Oh, actually, so we already have a question, which is great. There's a question about what is the refractive status of guinea pigs in their normal outdoor habitat? And are there natural variations in refractive errors? Sally?
Yes, no, indeed. Actually, the guinea pig, you know, it's a little tiny creature that sits on the floor, the ground, and it's always looking for predators above as well. So it actually is hyperopic in the part of the eye that is looking at the sky, and myopic in the part of the eye that's looking at the ground, and that's pretty common for a small mammal that lives close to has short legs and lives close to the ground. In fact, even humans have some lower field myopia as well. When it's much more modest than what you would see in mammals like that. So normally the guinea pig is emmetropic, though, in the central vision, and it develops just like any other species, including humans, where it emmetropizes and becomes, and so its refractive error ends up being normally in a normal environment, not having a refractive error, unless you start to do these experiments where you interrupt that vision with a diffuser or with lenses.
Great, thank you. We also have another question that's on yoking. So the question is it normal to treat in guinea pigs just one eye with intervention and leave the other eye as control? Aren't the two eyes dependent on each other?
Yes, well, that depends a lot on how much binocular vision individual species have. And so the guinea pig does have a binocular field, and there is some interaction between the eyes. But in general, if you compare that fellow eye that is a control, it's actually better to control the two eyes within the same animal than it is to try and look at different sets of animals, because the two eyes are so heavily linked together in terms of their refractive development. But in reality, the fellow eye looks the same as animals that don't have any diffuser on them at all. So they're pretty independently controlled in the guinea pig. Once you come to a monkey where there's a much bigger binocular field, then usually the experiments are done binocularly so that we can get around that problem of whether you've got influences from one eye to the other. And the role of binocular vision and convergent accommodation and how that interacts is an active area of research and is of interest in its own right.
Great. Thank you so much, Sally. Are there any other questions? If not, I have some of my own. So I noticed that Dr. William Miles is also here as well, and he did some of the work, as you pointed out nicely in your talk. I was actually quite intrigued by all the form deprivation and the image analysis that you did in terms of outdoor versus indoor. What do you, Sally or Will, do you have in terms of thoughts on how this has implications on urban design? Like how can we change things besides just telling kids to go outdoors?
Yes, well, changing behaviour, of course, is very hard, right? So we really need to change the design of the things around us rather than try and change our behaviour, because no one's going to give up their phone. And I did put in an extra slide to do with that. And Will, you might like to talk to this slide and explain to the audience what this is about.
Yeah, so essentially, when I did the image analysis, I was looking at how much regional exposure our retina would receive that would be equivalent to some of our manipulations that induce very significant myopia in our animal models. And overwhelmingly, the amount of our retina that would receive form deprivation or effective form deprivation increased dramatically when we're indoors, when we're in urban environments. And that's purely because we don't put any detail into our surroundings. We love clean walls. We love very clean spaces that look very, very nice and comforting. But to our eyes, it's essentially the same signal as what we give our guinea pigs. And in particular, it increases towards the periphery in our retina because when we're indoors, we're limiting the distance of space that we have in our periphery. So we force a lot of low spatial frequency detail close to us, but also the retinal sensitivity of the sensitivity of our retina to spatial detail decreases towards our periphery. So we've got this slide here. So you have a very good environment, which is this excellent example where the contrast exposure in both the periphery and the central vision is very high. That's very good. It's all very nice detail. And then we move to this pretty good example where we're focusing on a phone, but we've got this very nice detailed surround, which means that both our central vision and periphery still get some good contrast exposure. Then it gets a little bit worse when we start restricting the distance of our viewing as well. In this example, where we're still in nature, but now we're looking at a phone and we're sitting quite close to the ground. And then there's this absolutely terrible example where we're indoors and there's no spatial detail around us, and we're focusing our vision on this tiny little thing in front of us, and our periphery is just getting massive exposure to the same signals that we give our guinea pigs to send them myopic. So essentially, we can do things to interrupt that deprivation in the periphery by redesigning our urban settings, giving more detail in our environment, whether that be wallpaper or just textured materials. So rather than just having a nice blank white wall, using textured woods or things like that actually have some more spatial detail in them can improve that. So yeah, there's plenty of things to think about. Singapore's pretty good. It's got a lot of green over its buildings, but again, if you don't really look at it, it won't have much of an effect. Yeah.
Yeah, so I think, maybe we could redesign our urban spaces as well as our indoor environments, because our indoor environments are the worst of all.
Yeah.
And so, perhaps perhaps this could be the time for the wallpaper comeback or something, because it is that spatial detail that you put into your periphery that is important. But it's not just that; it's also the wavelengths of light. And also the lighting environments that we're using within our spaces, which are important. And there are certainly types of design within homes, within your own home, that would help dramatically in terms of the kind of signals that you're putting onto your peripheral retina.
Great. Also, there's three other questions now. The first two is from the same person. Does the time of day exposure matter for myopia, given the robust diurnal rhythms in axial length and choroidal thickness in animals?
Yes.
But then also the second is, what are your thoughts on the choroid and myopia in general?
On the choroid. Yeah, well, no, the time of day definitely does. The time of day is quite important because the eye, for a start, the eye grows during the day, right? But it also tends to grow most in the afternoon. And the inhibitory choroidal responses, where they are maxed out, is also important, right? Because we don't really want to. What's more interesting than the excitatory signal that's causing the myopia is actually the inhibitory signals. When are they most dominate? And so in the human studies now, there's been, because OCT has allowed us to measure the choroid in humans, and because we can now detect the choroidal changes and detect those circadian cycles, we can use that as a way of knowing whether or not the right signals might have been in the retina and are being pushed into the choroid, because they seem to change the thickness of the choroid. And so it has been used as an indicator of whether things are working or not. And that can be used to know whether, one lens or another lens might be working or not as well. But it's not the case that those choroidal changes always correlate with the eye growth changes. And so although it's not a bad measure, and it's the best we have at the moment, it's not the fail-safe measure. And we can't obviously go into human eyes and start measuring levels of dopamine, etc. And so it is a good measure for the time being until we get some more accurate types of measures. In terms of the diurnal cycles, absolutely, they are important. And it may be that the manipulation of those diurnal cycles and circadian rhythms is can help inhibit myopia for sure. Having said that, I think the way the eye works is that it's not just the circadian cycles, but there are multiple pathways that the eye modulates its eye growth. And the circadian pathway is one of those pathways. But there's also the rod and cone pathways that are sensing both contrast and spatial frequency and the wavelength of light and interpreting those spatial contents of our visual images. And so we've got these multiple pathways that are acting together in some way. So if we start manipulating the circadian rhythms, at least that's part of the story. If we can also manipulate the visual signals at the same time, then they will presumably summate together and we can get a better effect size.
Great, thank you, Sally. Sort of related to that is a question about red light therapy, actually. If there have been many red light therapy studies in small animals that you know of and their effectiveness.
Well, actually, there's very few red light experiments done in small animals at the moment. And so that's an active area to try and work out what is going on. Exactly where is that working? Is it changing the metabolism of the choroid? Is it perhaps working through those through the melanopsin, turning off the melanopsin pathways through those IP-RGCs is another possible pathway that it could be working. And it's possible that it's also modulating things in the sclera. So no, we have no idea really exactly what's going on in the animal models. People are doing those experiments now, but at this point in time, we don't have the answers. And it's certainly of great interest. What we really want to know is what would be a safe version of that therapy, and make sure that that we're not causing damage to the eye. If we have very high levels of red light, that's not good, just like high levels of blue light is certainly not good either. And what is this thing about, how come blue light can inhibit and red light can inhibit, how does that all work? And that's because we're talking about different pathways. So knowing how they interact is going to be pretty important. But the data that's coming out on humans on red light therapy looks amazingly impressive. And so there's a lot of interest in this at the moment, and the studies are ongoing.
Great. Thank you so much. Are there any other questions? If not, I'd like to thank Prof. Sally Mcfadden and Dr. William Miles for lending their expertise. And again, thank you everyone for your time tonight. And then just housekeeping wise, I think the feedback form will automatically be sent to you at the end of this webinar.
Thank you very much to the audience for taking the time to listen to what I had to say today.
Okay. Okay. Have a good night.
Bye. And thank you, Donnie, for hosting the session.
No worries. Thanks for doing all the work.
No problem.
Okay, I'm going to run down. I heard some screaming downstairs.
Yeah. Oh, okay. Bye.
Thank you, Sally. Bye.